Okay, great. Thanks, Joe. So we are very delighted, Karen and I are very delighted to have this opportunity to talk with you about our EDSR project. And so I'm going to talk for the first 20 minutes or so to give you an introduction to the kind of bioinformatics infrastructure that we have developed that allow us to build knowledge network. That then Karen is going to demonstrate how we use those knowledge network then to overlay with the omics data for interpretation to ask some scientific questions and interpretation of the underlying omics data. So I'm going to focus the discussion on well, basically the infrastructure that we have developed both at the Center for Bioinformatics and Computational Biology at University of Delaware, and also the infrastructure we have developed as part of the protein information resource located both at the University of Delaware and Georgetown University. And the specific area of focus of discussion today is our text mining approaches that allow us to build a very rich knowledge network. Okay, so PIR, so the protein information resource is a resource that we have developed over several decades that provide an integrated environment for genomic, proteomic, and systems biology research. And the special area of research is focusing on protein structure function in the beginning, but then we expanded that to omics data integration and visualization, and bringing a lot of expertise in biomedical text mining, and also engage in the community in building biomedical ontologies for our systems biology and more translational research work. And so we have built a quite solid bioinformatics framework, particularly for data management and analysis, and have been engaged with a lot of community standards development. And we are part of the national and international collaborative networks. Particularly, we are part of the UniProt consortium. We are also part of the Oval Foundry and leading the reference ontology, which is protein ontology development. We also have close engagement with the text mining community as part of this BioCreative consortium that provide critical assessment for text mining efforts. And so UniProt, as many of you probably are aware of and probably have been using as a resource for protein sequence function type of analysis to get rich functional annotation. So at the UniProt consortium consists of us at Protein Information Resource, but also our partners at European Bioinformatics Institute and Swiss Institute of Bioinformatics. And so collectively we've been doing certainly rich sequence and functional annotation, and this is based on both the literature-based manual curation, but also to help scale up the automatic annotation. We've been adopting rule-based, classification-based type of annotation approaches. One area that we have been focusing on is to quite relates to the LINKS project, is that we have been annotating a lot of functional features, and in order for us to present the functional features to the broad community, including the genomic community, we have engaged in mapping the protein features to genomic coordinates, and so that the sequence features could be directly mapped and used to do functional annotation of clinical genetic variants. And so this is just an example of how those functional features could be directly viewed on a genome browser for people to look at genetic variants and how those genetic variants may be interpreted based on their functional features, including structure functional features and active sites, functional sites, protein modification sites, and so forth. At the Center for Bioinformatics and Computational Biology, We have also built quite a bit of informatics infrastructure and also working across campus in establishing our educational programs. And one special area of focus of our informatics infrastructure development is in the support for next generation sequencing data analysis. And so it supports various applications for the NGS from the de novo genome assembly to RNA-seq data analysis to metagenomics and so forth, and epigenetics analysis. But the focus I'm going to have today is to talk about the specific effort that we have in the text mining area that allow us to go from literature to knowledge and allow us to build the so-called knowledge maps. That would be the focus of Karen's presentation in how that could be used, leveraged to interpret omics data. So for the literature to knowledge workflow, the first part is text mining workflow that consists of different modules. The modules that allow us to tag named entities, allow us to extract information from the scientific literature, including the relationships of those entities and the biological context of those entities. And also has this component for normalization that allows us to map those entities extracted from the free text to database identifiers, to ontologies and control vocabularies. Therefore, it would allow us to provide integrative analysis and discovery. And so this is presented in a semantic environment where the data could be visualized and could be analyzed basically in the context of data from other disparate resources for further hypothesis generation and knowledge discovery. So we have developed a number of text mining tools, and we have brought together those text mining resources in the PIR framework, where we have this hyperlink as a portal to several text mining tools. This website here presents the more mature tools, where it have gone through very extensive benchmarking and also user testing. And so from this portal, we provide the web user interface to the different text mining tools. We also make available the literature corpora associated with the development of the different text mining tools. We have several other tools that are currently under development that I will be talking about briefly that will be incorporated into this central portal as well. So this is a rather busy slide, but just to give you the very high-level ideas to the kind of things that we are developing. And so again, we have this overall workflow that starts from the scientific literature. We go through basic text processing. We have different kind of information extraction engine, the different applications that will allow us to extract different types of entities, different types of relations, different types of biological context. And then that would all be integrated for dissemination, integrated into databases, dissemination through web interfaces, through RESTful APIs, and so forth, and also for downstream analysis through graphical views. And the specific tools that I will highlight a little bit, the first one is Alliance PTM. And so this is a tool that allows us to mine post-translation modification information from the scientific literature. Specifically, we will focus on Alliance P, which allows us to mine the kinase substrate site information and the relationships. Then, because our particular interest in the consequences following phosphorylation, we have this EFIP that has a PPI module that allows us to mine specific protein interactions that involve phosphorylated forms, so that we can look at through these crosstalks between the phosphorylation networks. The next one is a mutation disease association tool that allows us to mine what's the disease consequence or association with genetic mutations or protein mutations. We also have a tool, Myriad, that allows us to associate disease with microRNA, including the kind of relations that leading a microRNA to association with disease, and MIRTAGs that allow us to mine the relationship between microRNA and target, and also genes that it regulates. So that's kind of the text mining tools that we are developing. We also, as I mentioned, because we are interested in building a pipeline that would be scalable and also would be quite interoperable with other text mining development efforts by the community. So we have been developing modules that could be used in our own pipeline and other people's pipeline. And one example is this iSIM for sentence simplification, especially when we look at the text mined from the abstract. Oftentimes authors are using very complex sentence structure that could be simplified and reduce the syntactic complexity of a sentence to multiple sentences that could be much more easily mined by using any other tools to mine the entities and mine the relations. We also, because we have been engaged in the text mining tool and system development for 10-15 years now, and have developed many different entity relation extraction tools. And so we have come up with this generalizable relation extraction framework where one can then take this generalizable framework to further develop new relation extraction tools for new types of entities and new type of relationships without having to build everything start from scratch. And with, for example, this iExtractor generalizable pattern-based relation extraction framework, we were able to develop the MIRTAG system that I will be talking about in a minute within 3 weeks. So we were able to build the system very quickly and go through then the benchmarking and further development. So anytime if there are questions, feel free to jump in. That would be fine. Okay, so the ALINSP, as I mentioned, it's a system for us to mine the kinase substrate site relationships, and so this is using natural language processing. And I just wanted to show very simply that from a tool, from a sentence that you extract from the literature, basically the NLP tool allows us to look into the different tokens, different parts of the part of speech, part of the language pattern, and then map that to some kind of pattern using the rule that would then allow us to basically convert the free text into structure format. And so, for example, here, using that kind of NLP rule-based approach, we will be able to detect the kinase, the substrate, the specific amino acid sites, and so forth, using the IE pattern illustrated there. This is just a workflow of the EFIP. Again, it shows this kind of modular approach where we can do is we take the output from ALINSP regarding the kinase and substrate and the phosphorylation site, and we attach another module that allows us to detect the protein-protein interaction for the phosphorylation sites, essentially the phosphorylated protein, and to identify that interactant that associated with the substrate, and that then further allow us to extract that relationship. And so once we have this kind of tools developed, then we can start bringing the multiple tools together to build network. And so this is an example of how just by bringing the ALINSP and EFIP together, we can already construct a phosphorylation network. And so this is an example that shows that we have two types of relationship. The P is the phosphorylation relationship, and so it shows that the RSK1 is a kinase that phosphorylates multiple proteins, the P27 and the BAT, and it modifies BAT at two different positions. And then it shows that the BAT proteins, upon modification, then it may have increased interaction with a protein like 1433, or a decreased interaction with another protein. And so that allows us to capture multiple type of relationships with multiple types of entities. And so this is a very simple illustration of what kind of PTM network that we will be able to build. But we don't need to start there. We can go from PTM networks to regulatory networks that involve many other major players in regulation, microRNAs, transcription factors, and so forth. And we can look at the different kind of entities that are important in the context of big data and precision medicine. Particularly, we are very interested in looking into the context of proteoforms and genetic variants. And so proteoforms meaning it could be the protein isoforms, could be the gene, the protein variants. We could be looking at different modified post-translation modification forms. We could also be looking at protein complexes with multiple subunits, and some of those subunits may be a modified forms. And we can look at them, this kind of different complex composite entities, also in the context of their in the context of disease and other biological context. And so the text mining tools that we continue to develop, in addition to the RPTM and EFIP, are listed here: MirText, Dymax, Myriad, and so forth. And we also have a PGen. It's a specific name detection and normalization tool for us to tackle proteoforms, the protein classes, and protein complexes. So this is an example, just a quick illustration of what MirText is doing. Basically allow us to extract the microRNA target relationship. And so it could be a direct target of the microRNA, or it could be an indirect target that is regulated by microRNA, or conversely, it could be a gene regulating microRNA. And so it could be, it could extract those different kind of relationships. And this is an example of once we have the kinase substrate relationships, once we have the microRNA relationship, once we have the transcription factors that are mined or could be integrated from the from curated data sources, then we can build this kind of regulatory network that covers those complex data types and relationships among them. So, so for example, here, as labeled, they include regulators for the PTM enzymes, and those PTM enzymes, the kinases, the phosphatases, are regulated by PTMs, by microRNAs and transcription factors. So we can start building rather complex knowledge network by integrating data from the scientific literature and data integrated from other curated sources. So Dymax, again, as I mentioned, it's a tool that allows us to detect the mutation disease association. So basically, it's looking into the genetic mutation, the mutated site, and the connection to different kind of disease. Myriad, microRNA in association with disease. So we know that many microRNAs are associated with disease, but in Myriad, we also extract the context. So for example, in this case, the microRNA 9 is participated in or associated with this particular cancer, but it's due to repress of E-cadherin and promotes tumor metastasis and so forth. And we have a BD2K software award that allow us to basically take this kind of text mining results and working with curators to further curate the results and use that for end users, the biomedical researchers, clinical researchers. And so in this context, we have many different type of entities and relations that we extract. And this is just an example of the kind of information we extract from this MACE2K text mining tool that will allow us to look into the drug, the disease, and the outcome information, and also the gene. And so in this case, it could be a gene, a kinase inhibitor that are used in the context of lung cancer, and what is the outcome information, say, extracted from clinical trial as reported in the scientific literature. So all this data are being disseminated. Again, we are part of the BioCreative consortium, where we have developed the so-called BioC exchange format to foster interoperability of text mining tools, text mining modules. So this is exchange format for annotated text. So essentially, any literature information, free text, annotated using any text mining tools, we could use BioC, we could use JSON format for data exchange, and so that people can decorate any free text with more and more entities and relations extracted by different tools. And certainly this is very much text mining in the BD2K context that allow us to really embrace the findable, accessible, interoperable, reusable, fair principle. So just very quickly, I think that's all I wanted to give in terms of the background. So now Karen will take over.
So I'm Karen Ross, and I work with Kathy. And as Kathy said, we're very pleased to have this opportunity to talk to you. And I'm going to now give you a specific example about how we used a subset of those tools that Kathy just described, together with omics data from the LINKS project, to look at the response of cells to kinase inhibitors. We, again, try to integrate biological knowledge, which we get from text mining and from other curated resources, with omics data, which in this case is coming from the LINKS project, specifically phosphoproteomic data and drug target data, and ultimately to answer some scientific questions that could be related to disease mechanisms, disease classification, therapeutic targets. And today we're going to talk about the relationship between kinases and the substrates they phosphorylate that you can see through the effects of kinase inhibitor drugs. So the knowledge resources that we used for this project were the phosphorylation-focused text mining tools that Kathy already mentioned, the RLIMS-P that can extract information about kinase substrate and site, and the EFIP, which can get the phosphorylation-dependent protein-protein interactions. And the results from these text mining tools, we've done large-scale text mining with these tools and integrated them into a resource that we call IPTM-Net. And together with the text mining results, we also have information from PTM databases that are high-quality, manually curated databases such as PhosphoCite Plus. And third, we bring in ontological information, particularly from the protein ontology, which is making a structured representation of PTM forms. And within IPTM-Net, we have a number of analysis and visualization options, and it allows users to look at PTM sites, PTM proteoforms, including forms that have multiple types of modifications on multiple sites, PTM enzyme substrate relationships, and also the phosphorylation-dependent protein-protein interactions. Just to show you a little bit more about IPTM-Net, one of the things that we provide is a sequence alignment view, which allows a comparison of PTM proteoforms within and across species. And we decorate the sequences with the different colored highlighting to indicate the different PTMs that occur at that site. The sequence alignment also allows you to predict by sequence similarity potential PTM forms in species that haven't been studied experimentally. And in this figure, those are the sites that are highlighted in gray. And more relevant to today's talk, IPTMNet also provides a network view. And this network view shows the relationship between kinases and sites, and also between sites and proteoforms, and between phosphosites and their interacting partners that are involved in PTM-dependent PPIs. So the LINKS project doesn't need any introduction to this group, but so we've been taking advantage of the LINKS data that is available from the LINKS data portal. And the two data sets that I've been using specifically are the P100 phosphoproteomic data and the kinome scan data that determines the effect of drug treatment on kinase activity. So the phosphoproteomic, the P100 data set, is a set of about 100 phosphosites that have been chosen to be representative of the phosphoproteome. And each site is part of a cluster of sites whose phosphorylation responds similarly across a range of treatments. And these sites were divided into 55 clusters, and then the 100 representative sites have then been assayed across many cell types and perturbations, and the data is available from the LINKS portal. And this is work that's been done in Jake Jaffe's group and was published in this paper that's shown down here by Avalon et al. So what we, in order to integrate this P100 omics data with IPTMNet, we took the 100 phosphosites that are used as the representatives in the P100 and looked into IPTMNet, which has kinase site information from all these different data sources, to see what are the experimentally known kinases that can phosphorylate the P100 sites. And of the 100 sites, IPTMNet had information about 16 of the sites. There were 16 cases where we had some information about kinases that had been shown to phosphorylate those sites. And it covered 15 out of the 55 clusters. Some of the sites had multiple experimentally shown kinases, so we had 26 kinase site pairs. And since we were looking at 100 sites, found information about 16, we had about an 18% hit rate of sites that had kinase information. And this is actually quite comparable to the rate that we've seen using other phosphoproteomic data sets. When we take a list of sites that people find in phosphoproteomic experiments, we usually see that IPTMNet knows about kinases for something around 15 to 20% of those sites. Since relatively few clusters were represented here, there's 55 clusters and we only had kinase information for 15, we decided to expand the pool of sites that we were looking at. So in the paper by Avalon et al., they actually originally had about 920 phosphorylation sites that they then divided into the 55 clusters and then chose the representatives for the P100. So we went back and took all 920 sites and then looked all those up in IPTMNet. And again, we only found kinases for about 18%, but now since we had a much larger pool of sites, we had about 170 sites that had kinase information, and it covered 46 out of the 55 clusters. And there were 351 kinase site pairs. So now we can build a network that shows kinase site relations for these sites that were clustered in the P100 and where we know kinase information from IPTMNet. So then, once we have this network, we can overlay it with the actual P100 data. So we chose a drug to work on, a kinase inhibitor called dinaciclib, and it's a CDK inhibitor specifically. And the experiment, the one network I'm going to show you is an experiment that was done on MCF7 breast cancer cell line with a treatment time of 3 hours and a dinaciclib concentration 4.5 micromolar. And when we took the phosphoabundance data for each of the P100 sites, either if the site was the site in our network was actually part of the P100, was actually one of the representative sites, we directly used that value for the phosphoabundance. For other sites that were from those 920 that were clustered, we calculated an average abundance for P100 sites in the cluster and then assigned that abundance to all of the sites that were part of that cluster. So the next data set that we brought in was the kinome scan data. So in the kinome scan assay, a panel of about 450 kinases is bound to ligand on a solid support, and then test compound is added, and then the assay measures how much kinase remains bound in the presence of the drug. So if something is an effective kinase inhibitor, it's going to displace the kinase from the solid support, and the results of the kinome scan are reported as the percentage of the kinase that remains bound in the presence of the drug. So a really strong inhibitor is going to displace a lot of the kinase, and you'll get very low percentages, whereas 0 is sort of the optimal complete displacement. For dinaciclib, we found the kinome scan assay available from Lynx had data on about 83 kinases that were in our IPTMNet P100 phosphorylation network. And the level of inhibition ranged from strongly inhibited all the way to not inhibited at all. And then you can see here the list of kinases that were strongly inhibited at dinaciclib with kinome scan values of less than 10%, and this includes a number of the CDKs, which you would expect given dinaciclib as being designed as a CDK inhibitor, as well as some other related kinases. So finally, here's the network, and the circular nodes are the phosphosites, and they are joined together into clusters. So you can see you can see that we have a number of different clusters from the P100 represented here. The kinase nodes are all represented as the triangles. Size of the kinase node indicates to what degree it was inhibited in the kinome scan assay. The P100 phosphoabundance is represented in the network as the color of the node. So the nodes where the phosphorylation was decreased in dinaciclib relative to DMSO are green, and the sites where the phosphorylation was increased in dinaciclib relative to DMSO are red. This close-up just shows a better view of the kinases, where you can see the ones that are more inhibited in the kinome scan are actually drawn smaller in the network. So MAPK9 and MAPK15 are two of the kinases that are quite strongly inhibited by dinaciclib in the kinome scan. So this is a subset of the previous network that just shows sites that were either phosphosites that were relatively strongly upregulated in dinaciclib, those are the red ones, or strongly downregulated in dinaciclib, and those are the green ones. And then the kinases that phosphorylate those sites. So just qualitatively, we saw that most of the significantly inhibited kinases, which are the ones that are really small nodes here in the orange circles, are phosphorylating sites that were downregulated in the P100. And so this was kind of a reassuring result that if you inhibit a kinase, then its target sites will be downregulated. And so by, and that was kind of borne out by when we put together these two sets of data on the degree of kinase inhibition and then also the change in phosphoabundance in the drug. Here we tried to plot that out a little bit more explicitly. So the horizontal axis here is the degree of inhibition of the kinase in the kinome scan assay, and the vertical axis is the degree of phosphorylation of the substrates of that kinase according to the P100. And every dot on here is a kinase. And the orange ones are the ones that maybe sort of behave how you would expect. These are kinases that are quite strongly inhibited by the drug, and also you see a reduction in phosphorylation of their substrates. The blue and the purple are two sort of interesting exceptional cases. The blue kinases are ones where they're quite inhibited by the drug, but the substrates, at least the ones that we can identify through IPTMNet, do not seem to be significantly under-phosphorylated. And then conversely, the purple kinases on the right are ones that are not really affected directly by the drug, but their substrates do tend to be less phosphorylated. So so why do we see these kind of the blue cases and the purple cases? There are some technical possibilities. The kinome scan is a little bit of an artificial context to look at kinase inhibition. So we're thinking of bringing in other kinds of kinase inhibition data. There's also kind of good biological reasons. Especially for those purple kinases like Aurora KB that were not directly inhibited by the drug, but we saw an effect on their substrates. These are kinases that could be indirectly affected by the loss of activity of a kinase that is directly inhibited. So for example, Aurora KB is a mitotic kinase. So when you treat the cells with dinaciclib, you lose CDK activity, which could reduce the mitotic index of the cells, and then therefore you would see, as an indirect effect, less activation of mitotic kinases such as Aurora KB. So where we want to go from here is to develop an automated workflow that will allow us to produce these networks, to bring in the P100 data, to bring in the kinome scan data for any kinase inhibitor P100 experiment that's in the LINCS data set. And this would involve retrieving the data, the kinome scan data, the P100 data, and then for each different experiment, for each different kinase inhibitor and experimental condition, modify a node file that will put in the quantitative data, the degree of inhibition of the kinases in the kinome scan, and the phosphoabundance in the P100 data set for each experiment. And the structure of the network actually will stay the same for all these experiments, because we're going to we still have the same kinase site relationships that all came from the that all came from IPTMNet and the knowledge network. And the only thing that would change in this as you go from experiment to experiment is the omics data that gets overlaid. We some things that we hope to add to this analysis to make it easier to get insight for what's going on would be the ability to select which nodes to display, to show to go one level higher. Right now we're at kinase site relationships, but we'd also like to know kind of what pathways are these kinases involved in, and can we say which pathways are activated or inhibited in the presence of these drugs. And then finally, ability to highlight differences between experiments. And I'm going to try to show you. So can everyone see kind of a black screen with a network on it?
Yes, we can.
Okay. So yeah, so we've been working a little bit with a group at NCI Frederick with someone there named Daniel Watson, who has developed a tool called Panoramix, specifically for these kinds of cases where you have many different experimental conditions and you want to show differences between them. So this is the Panoramix representation that he made from our P100 kinome scan data. And the node colors and the node shapes and everything are similar to what I showed in the network in the slides. And we did 3 different time points of dinaciclib, the 3-hour time point, which is what I showed in the slides, and then a 6-hour time point and a 24-hour time point that, again, all came from links. And you can animate this view, and you can highlight the parts of the network that are changing significantly between time points. So in each, as you cycle through the 3 different experiments, you can see which clusters of sites seem to be affected most by how long the dinaciclib treatment was applied for. And so we're sort of hopeful that something like this could be used to help compare between the many different experimental conditions that are available from links. And then I just want to put up our acknowledgments. So yeah, so here's our team for text mining team and IPTMNet development team over here, and these are scientists based both at University of Delaware and Georgetown University. And then over here on the right is our funding. And that is all, and thank you very much for your attention.
Hi, Karen. This is Andrea. It's very exciting to see someone playing with phosphoproteomics on this level. It's really exciting. It's really where we need to be going with the analysis of phosphoproteomics. So my question would relate to both the fact that you have multiple kinases oftentimes that can be responsible and can be active for particular phosphorylation sites, and how you're representing those layers of the multiple kinases that may be responsible, and also how I see opportunities in this also to stress where data might be missing. So as you said, there's I think you said 18% of the kinases could be found. So I think that there's definitely information there that you can drive where knowledge needs to be investigated and experiments to be done to find these unexplored kinases or unrepresented kinases.
Yeah, absolutely. Yeah, that's something that I think about a lot, because, like I said, every time we look at one of these data sets, we get like this 15 to 20% number, and so you realize that there's 80% of these sites where no one has really shown in an experiment, you know, that we can find what the kinase is, and so there's clearly a huge amount of work that can be done there. And your first point about the multiple kinases. So at the moment we're doing a very simplistic thing, which is if within IPTMNet, if multiple kinases are listed for a certain site, we will just put all of those edges into the network kind of with equal weight. Of course, different kinases are active in different biological contexts, and so something that, you know, could happen down the line would be to actually take that into account and pay attention to, well, you know, based on the type of cell this is coming from, even though IPTMNet tells us there's 5 different kinases that could phosphorylate this site, these 1 or 2 kinases are the ones that are most likely to be active in that environment, and that would add that would help a lot, I think, in focusing on what the most relevant kinase pathways are for the particular experiment.
Well, if there are no more questions, thanks again, Karen and Kathy, and everyone else for attending our data science webinar. And till next week.